Okay, so hello everyone. I'm Sarah Dunn. I'm the manager of the high throughput analysis lab. We're a core facility located on the Evanston campus, but we work a lot with people on the Chicago campus. So we are organizing these user spotlights to get the word out that we exist and also to give people a chance to show their work. So I'm really happy to present Euan Parnell. I've been working with him since 2016. I know at this point he's kind of an expert on half of the instruments we have down there or up there. And so he's going to be presenting some work he's doing identifying neuronal deficits in iPSC-derived model of schizophrenia. Yeah.
Hello, everyone. Thank you for coming. So I'm Euan Parnell, and I'm a research assistant professor with Professor Penzies in the Department of Neuroscience and the Center for Autism and Neurodevelopment. And I'm really excited today to give a talk in person for the first time in 2 years, and also about a project that I'm really invested in, really passionate about. And so I'm going to talk today about finding out what alters in neurodevelopment using a stem cell derived model of schizophrenia. And so just a quick background on iPSC, induced pluripotent stem cells. And so these are a really useful tool for modeling neurodevelopment and many other disorders. And so the benefit of them is that you can take fibroblasts or blood samples from a diverse cohort of patients for any genetic disorder you're interested in. And by introducing Yamanaki factors, you can turn these into a pluripotent state. And so these pluripotent stem cells can then be differentiated into almost any cell type of the body, which gives us an incredible tool to model tissues that we can't get access to and these diverse genetic backgrounds. And so we can model diseases with very heterogeneous backgrounds. So animal models are normally limited to a single mutation to find out what that does. But in fact, many different mutations can converge to produce disease. And so iPSC allows us to recapitulate all that genetic complexity. It also gives us access to tissues, human tissues that we can't get otherwise. Typically, we're interested in neurons in the lab. To get the kind of neurons that we're interested in, which are prenatal, developing fetus brain, it's very hard to get access to tissues like that. And even if we did, we don't know what their diagnosis is going to be. We don't know what genetic mutations that they have. And so with iPSC, we can model neurons or any of these other tissues without having to get postmarten samples. And finally, they're human. And so something I'm really interested in is drug discovery and development. And there's this huge rift between preclinical models. So many drugs are successful in animal models of disease, but then when you go into the clinic, they fail. And so a big push in the field is to try and incorporate human models of disease to try and improve those outcomes in drug trials. And iPSC are a route to achieve that. So one other useful thing is that when iPSC are in this pluripotent state, they can also be expanded almost indefinitely if you've got good cell culture technique. And that allows us to use these genome editing tools like CRISPR-Cas9 to modify the genomes from these very genetically variable patients. And so there's two routes that we can do that. We can take someone with a genetic disorder with a known mutation that we're interested in, and we can correct that using CRISPR-Cas9 to see if that is necessary for the outcomes, the neurodevelopmental defects that we see in our iPSC. And conversely, we can introduce a risk factor into a healthy patient to see if that is sufficient to bring about changes in how these neurons are developing from iPSC. And so I'm going to talk about these isogenic lines in the talk, and we've used the top route to show the sufficiency of the mutation involved in schizophrenia. And we've introduced this genetic risk factor to try and model schizophrenia. And so iPSC can be turned into many different tissues in the body, but in the Penzies lab, we're really interested in neurodevelopment, autism, schizophrenia, epilepsy, intellectual disability, all these different kind of genetically define models. And there's two routes that we can get neurons from this. On the top, we have the classic way, which is regulating growth factor pathways. Normally, there will be dual SMAD inhibition, and that can generate cortical neurons. But also by tweaking this, you can get a range of different tissues like microglia, astrocytes, dopaminergic neurons. And interestingly, if you grow these without attachment, they spontaneously form these spheroids, and these start to recapitulate some of the complexity of the brain in terms of the cell types that are there, and also start to recapitulate structures within the brain. And these are really interesting, but they come with some cons that limit their use. So they're take a long time to get a level of maturity to get that brain complexity. And they're also a complex system. So there's lots of different cell types interacting in these. We're trying to find out how genetic mutations can bring about changes in neurons. And so we were really interested in this directed differentiation, the second route. So in a landmark paper in 2012, the Sudoff lab found that by introducing a single transcription factor, neurogenin, into iPSC, you can drive the differentiation into a very pure culture of cortical glutamatergic cells. And so that was interesting to me because these are very uniform cultures, which makes it much simpler to find out the effects of mutations. It's a pure culture in terms of both maturity and the cell lines that are there. And it's very rapid. And so we can do more studies, we can do more interrogation of the changes that are happening during neurodevelopment. And so we have this model, we can start to think about disease. And we were very interested in schizophrenia as a neurodevelopmental disorder. And it's highly heritable. And so studies have shown that about 50 to 90% of your risk for schizophrenia comes from your either inherited mutations from your parents or de novo mutations that occur in your germline. And in fact, most recent studies suggest closer to 80%. So quite reproducibly, about 80% of your risk is inherited. And so it's a great model to study with stem cells. And here on the graph, we have the kind of effects of that inherited risk. And so on the left here, you have all these common variants, which confer very low risk, but there's huge numbers of them. Some estimates are up to 10,000 single mutations can contribute towards a risk for schizophrenia. But on the right, you have these either inherited or de novo mutations in single genes or in large portions of the genome that confer much higher risk to schizophrenia. So you have the 22q11, that's a famous model for schizophrenia that gives 65 times the risk for schizophrenia. But it's very rare in the population. Not many people have that. And so you might be thinking, we're using iPSC to model early development, early neurodevelopment, prenatally. Is that suitable for studying schizophrenia? And so you might think age of onset schizophrenia, 15 to 30 is typically when you get a diagnosis. But even before that initial psychosis, which is required for schizophrenia diagnosis, you see changes in neurodevelopment. So there are negative, these are mood disorders, and cognitive defects that occur even before the onset of disease. And this has been shown by both brain imaging studies. So there are changes in how the neurons are firing, the oscillations within the brain, even before a formal diagnosis and during development. And you get decreases in IQ in patients during childhood. And all these support an early developmental defect in schizophrenia that is suitable to study with iPSC. And so there's two ways to try and get at what is changing in the neurons of schizophrenia patients during development. You can generate these huge cohorts, and people are doing this, of hundreds of patients. You generate iPSC from them and try and find out consistent changes regardless of what risk factor they have. And you might get an idea of some consistent alteration in schizophrenia, but there's no guarantee that you're going to find that thing. It could be one mutation causes alteration in inhibitory cells, one causes a change in the function of excitatory cells. And so finding that consistent change is very difficult. And so a lot of studies using iPSC focus on these genetically defined models. And so you take one of these risk factors that is known to to be a positive factor in schizophrenia and try and find the changes that both may contribute to schizophrenia, that correlate with these larger studies with lots of heterogeneous patients and with actual patient live and postmortem studies. And so in our lab, we took on the task of studying the 16p11.2 duplication. So you can see it pointed out on the graph here. It is a relatively common of these copy number variants, CNVs. It occurs with about 1 in 1,000, 1 in 5,000 in the population. And it confers 10 times your risk for schizophrenia. So it's a really interesting model. It also confers risk to these early developmental disorders. So diseases with an age of onset around 2 to 5, intellectual disability, epilepsy, autism, further supporting iPSC as a viable approach to study the neurodevelopmental changes. And so what is 16p duplication? It's a copy number variant that results in a duplication of this segment of chromosome 16. And so 30 genes in this region are duplicated, meaning there are now 3 copies of each of these genes in the genome. And that seems to be enough to cause a really large risk for schizophrenia. So our goals for this talk are to define the neurodevelopmental alterations that happen due to the 16p11.2 duplication, correlate these to the phenotypes present in schizophrenia lines, and then correlate that with patient outcomes, patient presentation. And one of my goals is to try and find druggable pathways that are suitable for pharmacological intervention. And that would allow us to not only define the key genes in that large region of DNA that's altered, but also try and propose therapeutics that might be useful in treating patients with this. And so we started off this study using this Crispr engineered line. And so I mentioned earlier about these isogenic lines that we can use. So this was quite a mammoth feat by a group at Harvard, the Talkovsky lab. And what they did was that they used CRISPR-Cas9 to introduce the entirety of those 30 genes. And so CRISPR is usually limited in the size of the thing that you can either introduce or remove. And so this was a really amazing tool to get our hands on. They did extensive validation and by CNV analysis and transcriptomics. And they were happy to share this with us. And so we're really grateful to them for giving us a chance to study the 16p duplication. So this is an outline of our differentiation protocol at the top. And so we see that we can force the expression of this transcription factor, NGN2, and we get a really striking change in the way the iPSCs look consistent with neuronal differentiation. On the left, we have these very dense clusters, which is characteristic of iPSC. They're positive for stem cell markers. But just by introducing that single transcription factor, over the 7 weeks, they turn into these very complex neuron-like cells with dendrites and axons. So here we've labeled the axon initial segment with anchorin G and dendrites with MAP2. And we can see that it has normal axon dendritic polarity consistent with the neuronal lineage. We counted how many of our labeled cells, we labeled them with GFP, we counted how many of them have this neuronal marker MAP2, and over 98% of the cells that we generate using this method are neuronal. So that gives us a lot of confidence that we have a great model to study the changes happening in the schizophrenia model. And in the Penzias lab, we're really interested in dendritic spines, and we're really happy to see that we get really mature stem cells with dendritic spines. And so on the right, we have blue FP just to show the outline of the cell. And then we have these spines are MAP2 negative, and they have PRT2, which is a 16p gene, and it's involved in synaptic vesicle transport. And you can see synaptic vesicles moving down into the dendritic spine, into the synapse, and potentially being involved in cell-to-cell communication. But we wanted a way to study the effects of these mutations in a longitudinal manner And so we use this multi-electrode array. So these MEA plates are non-invasive. We can record repeatedly all the way through development. And we get this output, which is this fireworks show of neuronal firing. And so every time a neuron on one of these electrodes on the plate fires, we can record it. And so we get this output on the right at the bottom, which is a raster plot of all these events. And we put a black line wherever there's a single electrode event. And we're also going to talk about single electrode bursts, which is where one electrode will have very rapid firing. And then we have network bursts, which is where multiple electrodes in the culture fire simultaneously, which is suggestive of long-range connections between different neurons in the culture. And you can see as the neurons develop, they go from this very few network events to a very highly mature state where there's this oscillation where most of the events are in these network bursts. And that's really pleasing to see. We're getting mature neurons. And we're also going to talk briefly about the synchrony index, which is the likelihood that different events on different electrodes in the cover slip will fire simultaneously within a certain 20 milliseconds. And so I'd like to give a shout out to my collaborator and co-author for this project, which is Lorenza, who developed this project, and to Dina Simkin and Evangelist Skinners, who gave us access and support for using this technique. And it gave us really nice results. And so in our isogenic lines, we saw a really striking effect on the development of our neurons culture. So in the top, in the blue, we have our control lines, which don't have any modifications to the genome. We see they develop over the 7 weeks that we culture them. They fire more, and they fire in more network events. So the synchrony index increases over the 7 weeks, and the network bursting. We see a really strong deficit in both the firing rate and the formation of these networks due to the 16p11.2 duplication. And this was data produced by Lorenzo. And so we see really strong deficits at 7 weeks in vitro. We see mature neurons that are changing their activity. And so we wanted to try and get an idea of the molecular pathways that are altered due to the 16p that might produce these phenotypes. And so we did RNA sequencing. We took the RNA from our iPSC-derived neurons, and we measured how many of all of the RNA in the cell there is. And so we have, what was really pleasing to see is that all of the 16p genes that we introduced and that are known to be neuronally expressed had a 1.5 fold increase in the expression, consistent with an extra copy in the genome. And it was a really striking effect. You can see really clearly the duplication is there, and it's altering the expression of all of these genes. And so to get an idea outside of the 16p, what has changed in the neurons, we took our 216 deregulated mRNAs, and we compared this to lists of proteins and mRNAs with known function within the cell. And so by looking at the ontology, the gene ontology of our deregulated genes, we found that calcium ion binding was really significantly enriched. And so this suggests that all these proteins that are involved in calcium ion binding are altered in expression due to our 16p11.2 duplication. And with the help of Mark Forrest in the lab, we did some really interesting, we took that a little bit further. And so we generated a protein-protein interaction network map where each RNA in our dysregulated gene set was assessed for the protein-protein interactions that happen within the cell in terms of their actual interaction with other proteins. And using that, we can define networks. And it was really interesting to see neuron projection development, neuron projection extension, in addition to cell migration motility and chemotaxis, which in neurons supports a change in how the neurons are moving around and how they're developing in terms of their dendritic arbor. And so we went and we tested that. So we stained our cells at 7 weeks where we see all these changes in activity, and we saw a significant decrease in the length of their dendrites, which suggests that there might be a limited capacity to form connections with other cells within the network. But when we counted the number of synapses in puncta, a marker for synapses, we saw no change, which supports a change in intracellular signaling or synaptic strength rather than synaptic connectivity. And so we saw calcium ion binding to be really massively impaired in terms of the transcriptomics. And so we used calcium imaging to try and get an idea of what was happening within our cells that might contribute towards these network deficits. So this is just a short video of our calcium imaging. And so each blob in the video is a cell. And we measure the calcium levels using a fluorescence dye. And we can see every time the cell depolarizes, we get an influx of calcium. And we can count how many of these calcium events there are in a culture and across multiple cultures. And we saw a decrease in the frequency of these calcium events, which was really satisfying to see. We validated our change in activity using an orthogonal method. And we also can look at the synchronous events. And so at 75 seconds, I've slowed down the video a little bit, you can see 5 of these cells fire together within the network. This is a synchronous event. And so when we count how many of those there are in our culture, we saw a significant decrease, again supporting our MA analysis. But what's really useful about calcium imaging, and one of the reasons that we went to use that, is that we can look at the intracellular levels of calcium, the amplitude of the influx, how much calcium enters the cell following the depolarization, and how fast the cell recovers from that. And when we looked at that, we saw no change in the resting levels of calcium, but we saw this quite strong effect on the duration of our calcium events. And when we plotted out multiple events from multiple cells from multiple color slips and overlaid them on top of each other and compared them, we saw a significant increase in the recovery after a calcium event, which supports our transcriptomics. There's a change in how the cells are processing calcium information. And so, just to summarize, the 16p duplication by itself is enough to induce changes in neurodevelopment of iPSC-derived neurons. But how does this contribute to the schizophrenia presentation, the development of disease? And are these phenotypes relevant to schizophrenia? And to do that, to assess that, we took these patient lines. And so in collaboration with Jabal and Pablo Gaitlin in North Shore and the University of Chicago, we got access to this cohort of schizophrenia patients who had been genetically defined. They did CNV analysis and found that 3 of these patients had the 16p duplication. So this is an incredible find. It's very rare in the population. And so this is a great tool to try and find out if those effects of 16p are relevant and present in schizophrenia patients. So we started doing our MA analysis again. And interestingly, we didn't find a change in the firing rate. So there was no alteration in the mean firing rate between our 16p duplication schizophrenia sufferers and healthy controls. But what we did find was a really profound change in the formation of these networks. And so it seems that the change in firing might not be a contributing factor to schizophrenia, but this network formation is something really penetrant and really important to how these neurons are developing. And so we got a chance to do transcriptomics again. And so we did RNA sequencing on our 3 schizophrenia patients and our healthy controls, and we found out what changes in the expression of different mRNAs there are. And in kind of stark contrast to our isogenic line, there wasn't this strong enrichment in the 16p genes. So we only saw the significant increased expression of 8 of the 16p genes. Although all the genes showed in general an increase in expression, but that was highly variable, suggesting that the 16p causes an increase in the expression of these genes, but there's a lot of variability between patients. And so that can help us define what genes might be important in the phenotypes that we're seeing. And so we did our Go-ontology analysis to try and find out what pathways are changed within the cells. And we're really excited to see calcium ion binding, in addition to calcium-dependent phospholipid binding, to be significantly altered. So it seems that 16p, this was the only Go term that was altered in terms of molecular function, suggesting that calcium signaling is a really important factor in how CSTP duplication brings about changes in our neurons. And I've also highlighted that their cell projection. So we did the same protein-protein interaction network map, and we found all these different mRNAs that are involved in the morphology of neurons. And consistent with that, we recapitulated our decrease in neuron length in our patient lines. So the 16p seems to be causing a decrease in neuron length in schizophrenia patients. But interestingly, we saw a more robust phenotype in terms of the connectivity between cells. So we saw a significant decrease in the number of synapses and puncta. And that's a really common finding in schizophrenia. So postmarten studies, they find decreased dendritic spine density and the expression of markers relevant to synapse formation. And so it seems like other mutations, other factors in the schizophrenia patients might converge with our dendrite phenotype to produce changes in how the neurons are firing. And we did all our calcium imaging. I'm not going to go through it in so much detail this time, but we confirmed our no change in the firing frequency of our neurons, but we saw this significant effect on network formation by these two different measures. And what was really interesting was our calcium handling seemed to be a really strong deregulated pathway. And so when we looked at the duration of our calcium events in schizophrenia patients, we saw a significant decrease in that length, the time it takes for these calcium events to occur. And that went hand in hand with a host of other changes. So there was a significant decrease in the resting calcium levels and an increase in the amplitude of these calcium events. And so all of this suggests that 16p might change calcium handling, but there's a host of other factors within our schizophrenia cells that seem to exacerbate calcium handling. And so just to summarize these kind of results, to try and find out what's consistent, we see network activity by both Me and calcium is significantly altered in our isogenic lines and our schizophrenia patient lines. When we look at morphology, we see a decrease in dendritic length. And when we look at calcium handling, we see this consistent reduction in the event duration in all of our lines, which goes hand in hand with potentially convergent mutations in our schizophrenia lines. And so are these phenotypes that we're seeing consistent with schizophrenia? When we look at postmortem samples from schizophrenia sufferers, we see a really robust change in how these neurons look. And so when you measure the complexity of the dendritic arbor, they have less projections, they have less complexity, and they have a significant decrease in neuron length, supporting this morphological phenotype that we see in our isogenic lines and our schizophrenia lines as a potentially causative or contributing factor in these schizophrenia patient phenotypes. We can't do calcium imaging in patients. We can't cut open their brain and look at the calcium event duration. But what we can do is compare it to other schizophrenia models. And so on the top there, you have the 22q, which shows a really strong change in induced calcium. So you can induce depolarization of your neurons. And they saw a decrease in the amplitude of their event and a change in the recovery following depolarization. And when you introduce this one mutant disrupted in schizophrenia, another really robust genetic risk factor for schizophrenia, you see similar changes in the depolarization following. This is in a heterologous cell, but it seems that this one and 22q have like strong changes on calcium signaling. It might be a contributing factor to schizophrenia. So we can do calcium imaging, but a lot of people have looked at the transcriptomics, similar to what we did, but in postmortem schizophrenia samples, and they find that calcium ion binding proteins are really strongly deregulated, supporting our data. You can't get access to brain tissue, but you can get access to blood samples from patients. And so in this study, they took bipolar, or they did a meta-analysis of studies that had looked at bipolar disorder and schizophrenia platelets, and they found a change in the resting calcium in both of these disorders. And so all this suggests that calcium imaging, calcium handling, calcium activities really could be a contributing factor towards schizophrenia. In terms of network activity, this is something that's been really repeatedly and consistently found to be altered in the brains of schizophrenia patients by both fMRI on the right and EEG. However, we are looking at a pure culture of excitatory neurons. That doesn't recapitulate this kind of activity that we see in the brain. So typically, these EEG recordings in fMRI will look at these large oscillations of multiple cells. And schizophrenia typically presents with oscillation changes at very high frequencies, these gamma band oscillations, which we don't see in our pure culture of excitatory neurons. And these have been consistently linked to inhibitory input, so the action of inhibitory cells within the cortex, dorsolateral prefrontal cortex in particular. And so what we wanted to know is, can we start to measure the EI imbalance that we see in patients in our iPSC cultures? And to do that, we need to generate GABAergic neurons. And so Lorenza Carlotta, a very talented postdoc that I worked with for a long time, did this incredible study where she used a protocol published in 2017, I think, which can generate pure cultures of GABAergic neurons. So unlike the single transcription factor in our simple excitatory protocol, she introduced two transcription factors, ASCL1 and DLX2. But similarly to our excitatory culture, you get a very pure culture of GABAergic neurons. And these GABAergic neurons don't have any activity. We can't look at their firing. They don't have a lot of activity that we can see using multi-electrode arrays or calcium imaging. It's much more complicated. And so she decided to use a co-culture model. And so to try and model the brain, she used GABAergic neurons and excitatory neurons in the same culture at a 15 to 85% ratio, consistent with what kind of ratios we see in the human brain. And she did a lot of QC. She found that there was expression of GABAergic markers. The neurons expressed the neuronal lineage markers MAP2, and there was really high efficiency of differentiation. And at the end of our 7 weeks of culture, she found that there was no change in the number of GABAergic neurons present. And so functionally, we wanted to look at what these can do to our culture. So the first thing she did was RNA sequencing to confirm the expression of GABAergic markers. She sees the expression of somatostatin, calbindin, even a little bit of parvalbumin, alongside DLX1 and DLX6, which are all really strong markers that there are GABAergic neurons present in the culture. She took these co-cultures with excitatory and inhibitory cells, and just looking at the control lines, she determined what kind of effects that they have on a culture. And so we saw really robust changes in the duration of these network events, in network burst frequency, mean firing rate, number of random spikes. And so we can tell that these interneurons were really active in changing how the excitatory neurons fire within the culture. We saw these changes occurring at 5 weeks, which is consistent with publications on MEA and using patch clamp of the GABA switch. So the change in interneuron function from excitatory to inhibitory. And when we compared these phenotypes to a publication just last year, we saw all the same changes. So these interneurons are really affecting the culture in a very consistent way that is consistent with recent publications of interneuron activity on cultures. And they validated this with patch clamps. So we're really convinced that these interneurons are active and altering the activity of our excitatory neurons. So what do we see when we look at the 16P model? So this is just looking at our isogenic lines, so the duplication lines in our co-culture model. And we see a lot of the same things. So we see a decrease in the mean firing rate, which was also observed in our isogenic pure culture of excited neurons, and we see deficits in the network formation. But in addition, we see a change in the burst frequency, that single electrode bursting, which suggests that the interneurons, the 16p duplication interneurons, have some alteration in how they can regulate the network. And so we can just compare these changes. We see that our co-culture has consistent changes in mean firing rate, synchrony index, network burst formation with our excited neurons alone, but we don't see this burst frequency deficit in our excited neurons. And so both excited neurons and inhibitory neurons are contributing to potentially our schizophrenia phenotypes. But our conclusion for our upcoming publication is that excitatory function seems to drive this. So even in the presence of interneurons, we get a lot of the same deficits in how these networks are forming and the activity of neurons. And so we can summarize the findings. So 16p11 point duplication is sufficient to induce alterations in iPSC models of neurodevelopment. These phenotypes that we see in our isogenic line are also in our schizophrenia line and are very consistent with changes that are present in schizophrenia patients and in postmarten samples. And so we're really excited that we might have found pathways that might contribute towards schizophrenia. But does this help us determine the potential for treatment of people carrying this disorder? So what I'm really interested in from a drug discovery background is, can we isolate the etiological mechanisms, the genes in that large chromosomal duplication that are involved in these phenotypes? And can that help lead us to therapeutics to reverse the deficits in schizophrenia patients that might be useful to treat the disorder? And so to do that, we have to take our information from a couple of different sources. And so the first thing is using all the wealth of genetic data available to us from these large genome-wide studies. And then also we want to try and manipulate this using drugs. And so we cross-reference these findings with druggable targets. So, deregulated proteins in our 16p that have available therapeutics that we might be able to use for this specific treatment of schizophrenia and 16p11 point duplication. And so the real challenge is finding out what genes are involved in bringing about these phenotypes that we see. And so there's multiple different possibilities. It could be that one gene brings about all the changes that we see. It could be that multiple genes are required to bring about those changes. Or it could be that multiple genes produce multiple phenotypes that all converge to produce schizophrenia. So it's a really complicated system to interrogate, but we have some clues from genome-wide association studies. And so this single nucleotide polymorphism, so it's not in a coding region, and it's a single base pair change, but this has been shown to affect the expression of these three genes, TAG2, DOP2A, and MAPK3, which are within the 16p duplication, in addition to SES6L, which showed a strong trend, and KCD13, which is the closest gene. And interestingly, this single base pair change is enough to give risk to schizophrenia. It's not nearly as strong as the 16p duplication, but this suggests that some of the genes that are altered by this polymorphism may be important in the presentation of schizophrenia. And we looked at our RNA sequencing from our patient lines, and we saw all these genes were very consistently altered, which supports them as potential targets suitable for therapeutic intervention. So what do these genes do within the cell? So they actually all have a strong role in neurodevelopment, a well-characterized role in neurodevelopment. So in the top there, we have KCD13, which is known to regulate the ROA pathway. This is a GTPase signaling pathway, which has a really strong role in the extension of neurites, which was really cool, really exciting to see. So KCD13 can regulate the levels of ROA and then alter the dendritic arborization and the dendritic outgrowth within patients and within animal models. And it's druggable. So there are therapeutics available. So Fasudil has been in clinical trials. And rosin is another drug that can regulate this ROE pathway. And this has actually got a lot of attention recently in studies focusing on the 16p duplication. So this group just last year found that there were strong migration deficits in organoids derived from 16p patients, and that rosin could reverse some of these deficits. And so it's a migration deficit. It doesn't necessarily correlate directly to our findings, but it suggests that a pathway can be regulated and potentially used to target some of the deficits that we see. Type 2, tau kinase, has a strong role in microtubule stabilization, which is a key molecular pathway involved in neuron outgrowth. And so by phosphorylating tau, which you might have heard from Alzheimer's studies, it can actually stabilize these microtubules and promote neuron outgrowth. And again, there are small molecule inhibitors of type 2. And so we have a potential route to address our neuron outgrowth phenotypes in our iPSC model. In terms of MAPK3 and SEZ6L, these are two other genes in the 16p duplication, they lie in this really interesting calcium handling pathway. And so we have NMDA receptors and voltage-gated calcium channels, which in response to depolarization cause influx of calcium, which can activate calmodulin, CAM kinase 2, and then MAPK3. And MAPK3 has a role in synaptic plasticity by altering transcriptional and translational regulation. But this calcium signal is also nuanced by the activity of sarcoplasmic reticulum stores. So within the ER, there are calcium stores. In response to depolarization and calcium influx, you get calcium-induced calcium release. And SEZ6L has a well-documented role in regulating that process. Through phospholipase C epsilon, randomly receptor, you can increase the calcium levels within the cell in response to a normal depolarization event. And you see I've got little asterisks here next to all of these proteins. So in addition to SEZ6L and ERK, which were in the duplication, all of these genes have altered expression in our patient lens, which suggests that calcium handling at both the molecular level and on the cellular level are really altered. And in terms of therapeutics, it was really cool to see that trifluoroperazine is a ryanodine receptor agonist. So we see decreased levels of the ryanodine receptor in our 16p duplication cells. So we can activate that using this FDA-approved antipsychotic. And so it's a really interesting route to go down to try and alter some of these calcium signaling deficits that we see. In addition, ERK is involved in 50% of cancers. There are hundreds of ERK inhibitors out there, and so we have the potential to regulate some of these transcriptional and translational events using ERK inhibition. And finally, we saw a really robust change in the expression of synaptic vesicle markers. So within the cell, in response, neurotransmitters are translocated to synaptic sites in these vesicles, but to fuse to the membrane and to release their cargo, we have all this docking machinery. So we see synaptotagmin, synaptobrevin, SNAP25, RIMS, all of these are involved in docking the synaptic vesicle to the membrane, which is a required step for neurotransmission. And again, I've got my little asterisks next to all these genes, and we see altered expression of synaptotagmins, RIMS, complexins, SNAP25, in addition to the 16p genes DOCK2A and PRT2, which have a known function in regulating this process. And so how do we regulate this with drugs? So there are not that many drugs, particularly FDA-approved drugs, that target synaptic vesicle release. The one that I found was levetiracetam, which targets synaptic vesicles. But this typically inhibits synaptic vesicle fusion, and so it's unlikely to be of benefit in a model where we see decreased activity and decreased network formation. And so a more likely candidate for therapeutic intervention is these calcium channels. So the final step of vesicular fusion to the membrane is calcium influx, which causes the fusion of that vesicle to the membrane. And so we can target calcium channels using these compounds. So BAY K8644 is an agonist that can activate voltage-gated calcium channels, promote calcium influx, and then alter the function of these calcium binding genes, synaptotime and Dr. complex, and alter synaptic vesicle release. And interestingly, this radipine is a FDA-approved drugs for cardiovascular issues, heart disease, but it's also been in clinical trials for Parkinson's. And so it is blood-brain barrier permeable, and it provides a potential route to alter synaptic vesicle release within our 16P carriers. And so I'd love to show you that these drugs work, but that's a work in progress. We're still working on it. But what we do have is a really a whole host of high throughput techniques available to us that we can try and study the phenotypes that we see, but also the reversal of drugs to identify potential therapeutics. Sorry. So we have our MEA recordings to measure the activity of neurons. We have calcium imaging in high throughput. So using this fluorescence imaging plate reader, we can see calcium transients, network events within a culture, and in response to depolarization. Very pretty. This was a video made by Lorenzo, so Lorenzo collaborator. And finally, we have high content imaging, which is relevant to our neuron outgrowth phenotypes. And so we can measure the neuron outgrowth during development and see the effect of drugs in real time. And so that's really exciting. That was a 12-hour video, and these iPSC-derived neurons were 1 day old. And so you can see they start as with very small dendrites, but very rapidly extend out. And so we're hoping we can regulate that initial step with some of these ROA inhibitors to implicate some of these genes and some drugs. And finally, I want to give a shout out to Sarah, who did a really wonderful job with high content imaging. And so on the top, we have our manual acquisition. The technicians in the lab who were there did a fantastic job imaging and analyzing the morphology of our neurons, but it took them a very long time. They were working very hard to get this data. And Sarah, we gave her the cover slips, and in a week, I think, took hundreds of images, analyzed them all, and found exactly the same results as us. So she saw a decrease in neuron length, no change in synaptic number, and no change in the dendritic carburization, which matches our cells. And so we're really excited to use high content imaging to measure the expression of synaptic markers and other markers of schizophrenia and 16p11.2. And hopefully we can use that to confirm some of the long-term effects of our drugs in this system. And so you'll be getting a lot of cover slips from me. And so just to finish up, I need to thank a lot of people who helped with this study. It's been a really exciting and wonderful thing to work on, a wonderful place to work. So Peter Penzies came up with this project and has given incredible guidance throughout. I worked with Lorenzo Carlotta, and I can point out her face. This is Lorenzo. So she did all of these co-culture models. She developed the MEA system. She did an incredible job, and it's a real delight to work with her. She's now moved on to Technopole in Italy, but hopefully we can continue to work together. The technicians in the lab, Blair, Hiba, Dan, and Kat, who've done an incredible job maintaining our cultures, feeding them, keeping them in a really happy state. I want to thank Mark Dos Santos, who's in the audience today, and Lola, who taught me to do the calcium imaging on Christmas Eve 4 years ago, and really helped with the analysis to get some of this really important insight to this calcium handling deficit. And everyone in the lab and who helped out with this, Mark, who did the bioinformatics. It's been wonderful to get everyone's help to really progress this project. Jabao and Pablo at North Shore and the University of Chicago gave us these patient lines, this incredible cohort to study schizophrenia. The Talkowski lab in Harvard gave us these isogenic lines to find out the effects of the 16p duplication. And Sarah and Chihao have been really supportive all the way through this study and other projects in the high throughput lab. And so thank you all for listening. And I'd be really happy to take any questions anyone has about this.
Can I ask you a question?
Start with people here.
Hello?
Hello, so I can hear people. Who's that talking? I can answer a question.
Yeah, okay. I had a, can you hear me? Yeah, I had a question and a comment.
Yes.
So my question is, I mean, I think it's a really good idea to use human derived cells for doing experiments like this, but rather than doing what you're doing, why don't you just grow cortical organoids, which will put your cells all in the correct kind of juxtaposition with each other? You'll have astrocytes in there and everything. I mean, it seems that the experiment that you're doing is kind of the previous experiment to what people are doing now, that's all. And my second comment is that the data that you showed when you're looking at the effects of calcium influx on vesicle release, right? The drugs you're using, I mean, the thing is the drugs that you're using like irazepine and the BK8644, the calcium channels that they target are not linked to neurotransmitter vesicle release. So the reason that irazepine is in clinical development is because it will affect calcium spikes, for example, in the cell body, which could be important, but those drugs don't affect the calcium channels that are linked to vesicle release, of neurotransmitter vesicle release. So that experiment, I don't understand what the basis is and why you're doing that actually. So that's my comment, and the other was a question.
Yeah, so I think yeah, I completely agree with you. Organoids are a fantastic way to study neurodevelopment. It's a much better model to try and interrogate all these different cell types and how they might be interacting and they've got much better maturity. So I would love to do organoids, and I think it's been one of our plans for a long time to move into that. One of the benefits of so what appealed to me about the directed differentiation, this simpler model, is that we don't know the driver genes, we don't know what changes are happening within those neurons, even in a single cell type. And so one of the goals was to produce a very simple model that we can interrogate in a lot of depth. So it's faster, we can do more studies on it. But it's also a very easy way to find drug effects. And so we don't have to try and interpret the results of drug intervention. If we add a drug and the cell line responds, we know that all the excitatory lines, all the excitatory cells in the culture are responding the same way. And that, you're right, that we can't extrapolate that to how a brain would respond. And so our first step was to isolate driver genes, like things that we can target to reverse some of these phenotypes. And then we're hoping to take this into a more complex model, either an animal model or an organoid. And so I completely agree with you. I think there were some benefits of using the induced neurons in terms of the throughput and the purity in terms of getting consistent drug responses. But yeah, organoids, I've got a dream to move into that. So you preempted me. In terms of your drug question, yes, it was a simplistic way of presenting the data. Some of those drugs aren't going to be the way to cure the disease or reverse some of the phenotypes we see. It was largely based on the deregulated genes that we saw. And so, yeah, maybe irazepine is not going to be the best way to alter calcium transients or neurotransmitter release. But because those genes are implicated through our transcriptomics, we thought that was a good candidate to start on, not just looking at the 16p genes, but also the genes that are dysregulated in response to that. And so we're trying to make a list of drugs that are suitable for repurposing and to see what effect they have. And our hope is that using high throughput techniques, we can really test quite a few drugs to find out what ones have the most benefit. Yeah, yes, I agree with all of your comments. Yeah.
Okay, thank you.
So I'm going to answer some questions from the chat now. So I've got a question asking how many differentially expressed genes overlap between the 16p, the isogenic line, and the patient. So we got a significant enrichment. There was a significant overlap in the genes between the isogenic lines and the patients. But I was surprised to see that there wasn't a larger overlap. So I think we got about 30 to 50 genes that were similarly altered, of which 10 were 16p genes. And so that's a very short list for the same really robust mutation. Like we know that 16p gives a really strong risk of schizophrenia, but in our patient lines, we saw a very different set of expressed genes. And what my conclusion is one, there's a lot of variability between patients, which maybe limited some of our patient studies. It's not a huge cohort, but also looking at the Go terms that we got, so the lists of deregulated genes annotated by function, there was quite a large overlap, but in different genes. So for example, in the isogenic lines, we saw VAMPs, a presynaptic vesicular protein, but we didn't see that in the patient lines. And so there seemed to be similar molecular functions altered, but the response in terms of the transcriptomics was different. And so it could be that there's an initial event, an initial change in the function of the neurons, and different patients, different individuals respond differently to that, depending on the genetic background. And so we're really trying to work out what is the most consistent things. We think that's going to be useful for identifying these driver genes. But yeah, it was some variability. I hope that answers your question. Based on MEA data, the NGN2 induced neurons are matured how many days? So we mature our neurons, so most of our studies went up to 7 weeks in vitro, so 45 to 50 days. And we found that to be a great time point to study because just after that, the cells start to plateau in terms of their activity. They don't develop much higher activity or network formation. And so 7 weeks seemed like a great time to study the effects of the 16p because we saw really profound changes. But we have cultured NGN2 neurons, I think, up to 15 weeks now for some other studies. And so it was really determined by the phenotypes that we saw in the patients and where that seemed to be the largest effect. So the questions in the chat, does anyone have any questions in the room or?
Couple questions. So to try to reduce some of the variability in the patient lines, are you attempting to make isogenic lines for those, or how hard is it to correct that with CRISPR is such a big duplication?
So that would be the perfect way to, to try and find out what other factors are contributing to those different phenotypes. For people who didn't hear, are we making a corrected line of our schizophrenia patients? And so yes, I looked into that. So we defined the breakpoints in terms of we use whole genome sequencing and validated with Sanger sequencing. I know the exact point which we would try and remove that the second copy of these genes. And it just turned into a really extended process. It was going to be really hard. I mean, the Tarkovsky lab who gave us the introduced mutants, it took them 2 years, I think, to generate this model. And so it's definitely been in the back of my mind to do this. And I had organized it with some collaborators, but it fell through. If anyone is interested in doing some CRISPR-Cas9, I would love to get in touch. Yeah, I can give you my email.
My other question is more about the core. You mentioned a few different types of methods, but is only the last one done to the type of the core? And like, can you do kind of live cell stuff to the core? I don't know if that would work for me, so I'm here to understand.
Yes, so
you just do the fixed cover slips for the core.
So I have been taking fixed cover slips up because it's a real nightmare to carry up live cells to Evanston. I do have a plan to use their cell culture facilities. So we have frozen stocks of all of the reagents that we need. And so potentially I can use the confocal automated imager to get the same data, which is much higher quality. It's just some of the technical aspects are much harder. So the live imaging that we did was, just like, but it was upstairs in Antec. They have the INCI site, and I think the CAM, the imaging core across the road, also has one. I would love to do confocal because there was a real issue with focus. It was hard in this complex culture to get a really clean focus on the GFP labeled neurons. And so the confocal is the best way to go. But yeah, it's technically a little bit challenging to be very careful carrying them up the road.
We do have research that bring things up. Obviously, there's a question of what the trip does to them. If it's, for students, you probably could bring them up. He doesn't have to bring them up the day of the imaging. Before and let them rest. But the other thing he had was the flipper, which will actually be on this campus. We had a flipper used it. Lorenzo used it, but it's no longer working, but we have a new one that we'll be purchasing.
And I have a couple other projects, and I'm up in the throughput lab all the time. So,
Really nice talk, really nice story.
well, thank you very much.
So, is there a existing model?
So yeah, it's a great place to work. So Mark in the audience actually has done a really extensive study on the animal model. And so we're hoping to move some of the results from this correlated with the effects of the mouse model. One issue with the mouse model that was very attractive with the iPSC is that there are actually 3 or 4 different mouse models of the 16p duplication, and they all have different phenotypes. So there's no consistency within mouse models with only that duplication, which suggests that the background of the mouse is really heavily influencing how the mutation affects the progression of these neurons in the animals. And so iPSC, with our patient lines, we can actually see what of these factors are relevant to the progression of schizophrenia. And that was the interesting thing about using iPSC lines for this. It takes out some of this unknown with animal models. But yeah, I think if we find a drug that reversed the phenotype, if that phenotype was present in animals, then we would have a really good candidate to take into kind of like preclinical studies.
Is there one of the 3 ones that some patients, or is there variation between them?
A lot of them have been doing behavioral studies. There have been some morphological studies that show some similarities. But they often go, a lot of these studies don't use the exact same techniques that we have done. And so correlating it directly, we need someone to do some of these really in-depth kind of molecular and cellular phenotyping. And I think, yeah, someone might be doing that. Yeah, I don't want to give too much away. This is Mark's project, but yeah, so in the lab, we are studying both the animal model and the stem cell derived model. Peter has a real strong interest in the 16p. Yeah, Mark will hopefully give a talk some other time and can answer these questions better than me.
Okay,